It's Mr. Sinti, and I'm going to talk to you today about a really cool technique called polymerase chain reaction. And I wanted to start off a little bit talking to you about why you would even need to do something called polymerase chain reaction before I went about explaining how it's performed. And so, say for example, we were interested in determining the difference between two individuals. We could perform something called a DNA fingerprint. And so what we would do is we would take a little swab and we would pull out, for example, a little bit of the DNA from one person and another person. And so here's the DNA, like this, between these two individuals. And so what we do is there's known differences between individual B and there's individual A. And these known regions in DNA are known to be polymorphic, meaning many different kinds. And so one of the areas that we use in the lab is something called small tandem repeats. And so there's these little areas in DNA where there's known to be differences between individuals. Now, what's interesting about this is that before we perform a DNA fingerprint, in other words, run it through a gel, an agarose gel, what we want to do is amplify and make many copies of the DNA so that we could study it. And so what we're interested in, I'm going to use yellow here, what we're interested in is just small regions right here in DNA that are known to be different. And so what we're going to be doing is performing a technique called polymerase chain reaction, or PCR. And basically what this is going to do is make many copies of this particular area. So it's DNA replication. So we're going to make many copies of this. And then what we're going to do is put this into a test tube, all this DNA into a test tube. And then this may be familiar from a previous video. Then we're going to add restriction enzymes that are either going to cut or not cut these particular areas. And then we're going to run them through an agarose gel like this. We're going to add the DNA samples here, and of course this is positive and negative, and by the banding pattern that we see between individuals we can be able to determine if two individuals are similar or not. So we could use PCR to make many copies of a target region in DNA. That's one application. Another application is sometimes in paleontology, which is the study of fossils. Biologists are interested in pulling out DNA from an organism that's no longer alive. In other words, a fossil, remains of a once living organism. You could scrape like a bone or a tooth or a hair or a seed, any sort of remain from the past. And believe it or not, there still might be a little bit of DNA present in the bone. You're like, wow, it's very rare and there's just a little bit. I wonder what we could do. Maybe we could sequence it. Maybe we could sort of look at that DNA and analyze it. And so what scientists are able to do is make many copies of the DNA. And so the many copies of DNA is called polymerase chain reaction. Then we can study it. An application that we have at school here is that we're studying redwoods, as you may have heard. Students in the lab are able to extract DNA from a redwood tree. I'll go green with the DNA this time. So this is known as genomic DNA. Now, of course, if we're going to analyze DNA, we can't analyze the entire genome of DNA. We're just interested in a particular region or a particular gene. And so this is the cool thing about PCR, as I will get into in a moment. But we're interested in a particular target area. And so what students are able to do is, using PCR, is to make many, many, many millions of copies of this particular gene. And then what are we doing with that? We actually take that redwood DNA and we splice it into a bacterial plasmid, transform it into E. coli, and then we purify the plasmid. From that, we clone it, purify the plasmid, and then we sequence this particular DNA. It's pretty cool. PCR could also be used at a crime scene. Like for example, if a small drop of blood or a cell was left at a crime scene, one of the first things that you'd want to do is try to extract the DNA, and then again, like DNA fingerprinting, look at a particular area and amplify it so that you could run a gel. Or you could take a sample of someone's blood and you could amplify a particular gene and then run it through a gel if you're interested in doing a genetic test. So there's a multitude of applications. It's one of the most important. Of course, you have to moderate that because you know I'm prone to hyperbole, but I think others would agree with me on this, that polymerase chain reaction is one of the most important techniques that we have in biology. And this video is going to consider it. And so one of the first things that that I'm going to tell you today in this podcast is that we're going to be working with DNA that comes from a virus, and it's not to be feared too much because it's a bacteriophage, and bacteriophages are unique to infecting bacteria. But if you recall this, let me click on this so you can sort of take a look at this. A bacteriophage, these are bacteria, and so a bacteriophage comes along. It's a virus. It's made up of protein on the outside, and what these viruses can do is that they can attach onto poor unsuspecting bacteria. The protein stays on the outside. It's a very dramatic video right there. And then the DNA gets passed down through this body and it enters into the bacteria. And the bacteria is then destroyed as a result of that. And so we're going to be working with viral DNA. And it's like, why would we do that? Well, one of the reasons why we would do that is that it's easy to work with and it's readily available. So if I were to draw a bacteriophage like this, some tail fibers here like this, what's interesting to note is that the DNA inside the bacteriophage is linear, and I'm not positive on this, but I think it's around 25,000 base pairs in length, the total length of it. So it's not very large. But what we're interested in doing is targeting one little small area like this of the bacteriophage, and we're going to try to make many copies. And so in our test tube we have like this something. The phage is called lambda, the Greek letter lambda. It's called lambda phage. And in that test tube, we're going to have this big piece of lambda DNA. But we're not interested in making many copies of that. We're only interested in copying just this one little segment like this. And so here's my question. How are we going to do this? I made mention to it, like we're going to make many copies of a particular target sequence, but how is it that we're going to do that? So a brief cliffhanger. And so let's get right into this conversation right here. And so as I mentioned to you before, the technique is called polymerase chain reaction, and this will be explained as the discussion progresses. So it's PCR of lambda phage. This is a picture of the virus right here. And what we're going to do is amplify a small region of DNA from this virus, and then we're going to run a gel to see if we actually did this or not. And so one of the things, the terminology that we use in PCR is something called template DNA. And template DNA is the DNA that you're studying. And so this is coming from a virus in this case. But again, if you're studying redwoods, it's a redwood is the template DNA. It's the target area that you wish to amplify. And so how do you go about doing this? And so how do you get a particular area? And the way you do this is that if DNA looks like this, say this is the whole thing and it's double-stranded. So I'm not going to draw in the A, T, C, and G's between them, but it's double-stranded. Well, one of the first things that you have to do if you want to make a replicate DNA is that you got to get the DNA to open up. And the way you get DNA to open up in the cell, you may recall, is there's an enzyme called DNA helicase, which unwinds and unzips the hydrogen bonds. But as it turns out, that in a test tube, outside of a cell, what you could do to get DNA to open up like that is apply a tremendous amount of heat. And so I'm going to put a flame down here like this, but if you apply 95 degrees, so almost boiling, you can get the DNA to open up like this. And so that's an easier way to do it. And so when the DNA opens up, I'll just draw in the nitrogen bases like this. Now this is the whole piece. We're not interested in amplifying the whole piece. Say it goes off in this direction, in this direction. Say we're just interested in this particular target area. And so this goes really far in this direction and really far in this direction. So how do you amplify just this particular area? Well, first thing you have to do is heat it. And then the next thing that you have to do is add something called primers. Now primers are little pieces of DNA. Let me go green with the primers. So they're little pieces of DNA. And what these little pieces of DNA do is they bind to a small area in front of the target and over here on this side, on the other side of the target. And so what this does is this oligonucleotide searches out of all the DNA in the cell. It's quite remarkable. Out of all the DNA, it's able to find its complementary sequence. And so this is an A, this is a T, this is a G, this is a C. So it binds to this side and binds to this side. And so you need these primers to go in there. Once the DNA opens up, the primers are then added and it attaches. And then what you're going to do is, you might have guessed this ahead of time, you need the enzyme DNA polymerase, because the enzyme DNA polymerase is then going to attach new nucleotides and grow the DNA in this particular direction and in this direction. And so you're going to be able to amplify just this target region in DNA because of these primers. They're really important. If you're still a little unclear on this, it's okay. Perhaps it'll illuminate a little bit further in the discussion. But as I said before, these primers are typically kind of small. They're around 15 to 20 bases long, and they bind to the complementary region of the target region of DNA that you're interested in doing it. So they provide a replication start point. So again, if I can illustrate it, if here's DNA, I'm going to just go single strand on it this time. What the primers are, I'll go double strand. So like this, what the primers are doing is attaching here and attaching here. They're known as forward and reverse. Now, in this level class, we didn't really get too far into the anti-parallel nature of DNA, but what the molecules are in opposite orientation, and DNA polymerase can only replicate in one direction, in a 3' direction. And so what's going to happen is the primers are going to attach like this, and then DNA polymerase is going to come along and it's going to extend DNA in this direction and extend it in this direction. And so those primers are really important because they provide a starting spot. You get the target. So you need template DNA. In this case, we're using virus. Could be anything. Could be your DNA that we study. And in fact, in another course, we will be studying your DNA and doing a PCR. So you start off with virus DNA. That's the template. You need to add these primers, which are these small little pieces of DNA that attach right to the area that you're interested in. Now, mind you, let me animate this if you don't mind. This goes really, this goes really far in this direction. And I'll just do this like that, really far, and this goes really far. And so this is the virus DNA. But we're just interested in this one little target sequence. And as it turns out, you might, here's a legitimate question: how big is this? So this target sequence is about, in this case, 500 base pairs. It doesn't have to be this big, but this is where the forward and reverse primers will attach. And so when they attach here and here, then you're going to get only this target sequence to make copies. And as it turns out, in order for DNA polymerase to make copies, you need nucleotides, or else how will it do this? And so, again, this is not in a cell. This is in a test tube. So if you put in the template DNA, and then you put in your primers there, and you add DNA polymerase, and you heat it and open it up, you're going to need to add nucleotides to this as well, so that they have something to use to replicate the DNA molecule. I'll talk about this a little bit in detail in a moment, but the enzyme DNA polymerase, as it turns out, it's a protein, but it doesn't really work as efficiently as it should unless this little magnesium, I'm going to just make magnesium as Mg+2, magnesium ion. It's a positive cation, and it seems rather small compared to the huge protein. But when it attaches to the protein, let's do this, Mg2, it allows it to change shape and helps it to work. So that needs to be added. And in the cell, there's lots of magnesium, so it's a non-factor. But in a test tube, you need to add magnesium. You also need to add the buffer. As it turns out, the pH can get a little out of hand during the reaction, and so buffer is a chemical that helps to maintain the pH. And then, of course, you're going to need to add the enzyme itself. It's called Taq polymerase, and I'll explain why that's called Taq. It's a DNA polymerase, why it's called Taq in a moment. So these are the goodies or the ingredients for PCR. So that's important. So let's talk a little bit about the fact that temperature is a critical component to PCR. I mentioned before that in the cell you could use DNA helicase to open the cell, open the DNA, but in this case you want to avoid using as many enzymes as you can. You need DNA polymerase, but you could use heat. And so if you take the test tube up to 94 degrees, it will allow the DNA to denature or open up. And then if you cool it down to 60 degrees, in test tube I mean, so I'm going to draw a test tube over here. So if you heat it to 94 and then cool it to 60, the primers will have a chance to attach or anneal. This means attach to the template, to the target region. So the primers anneal, and then you warm it up to around 72 degrees, and that's when DNA polymerase can start elongating or replicating the DNA. And so then it's these three steps repeated over again, and then you'll have 2 copies of DNA, and then you'll want to heat it up again, and then cool it, and then warm. That's 2 cycles, and then heat it, and then cool it, and warm it. And so this is why it's called polymerase chain reaction, because it's a chain reaction of DNA polymerase replicating DNA over and over in different multiple cycles. And so the heat, again, 94, 95 degrees will get DNA to open up, and you need that. And then you want to cool it down to around 60 degrees for the primers to anneal. And there they are annealing down below there. Then the polymerase is going to need to attach to this area and this area. And that's why you're going to warm it to 72 degrees. And then the DNA polymerase is simply going to extend or elongate and replicate DNA. So that's what's doing it. DNA polymerase is replicating the DNA. And so here's a picture, cartoon drawing of DNA polymerase, and it's attaching to the primer, and it's searching for the A's, T's, C's, and G's, and it's replicating DNA. And so the reason that it's called Taq polymerase is that, I don't know if you would have caught this, but most proteins don't hold up very well in terms of structure when you heat them to 94 degrees, 95 degrees. That heat alters their shape and they usually don't function well. But interestingly enough, scientists discovered in these hot springs of Yellowstone National Park, they took samples of this boiling water in the national park, and they found bacteria living in there. So if bacteria live in this boiling water environment, that's a pretty extreme uh, situation. And so clearly they have DNA polymerase and they're able to replicate. And so what we did was isolate their DNA polymerase. And so we use a bacterial, I find this to be very interesting, we use a bacterial enzyme called Thermoaquaticus or Thermus aquaticus bacteria in PCR because it's sort of doesn't lose its shape in boiling water. So we could use heat to open up the DNA molecule. It's quite remarkable. Then I mentioned this magnesium. You just don't pour in the element magnesium, but you add magnesium chloride, the salt, and then it disassociates into the magnesium ion, and that helps DNA polymerase to work. Without that, DNA polymerase isn't looking too good. And then, of course, you need to add to the test tube the A's, T's, C's, and G's. Now, again, in this particular course, I haven't emphasized this, but I'm going to need to at this moment if you don't mind. Normally I talk about a nucleotide as a phosphate, a deoxyribose sugar, and one of the 4 nitrogen bases with A, T, C, and G. But the truth is that these A's, T's, C's, and G's, these nucleotides, are actually called DNTPs. So it's deoxynucleoside triphosphate, and the N stands for either an A or a T or a C or a G. It turns out that there's 3 phosphates attached onto the nucleotide, and when the last 2 are broken, it provides the energy necessary to attach the nucleotide onto the DNA. So these are known as the DNTPs, but basically it's just your good old A, T, C, and G, and those are over here. That's what this represents. It's either A, T, C, and G. And you could actually purchase these, and they come in the mail. They're the DNTPs, the DCTP, the DGTP, the DATP. And these are what the polymerase is going to use to extend the DNA during the polymerase chain reaction. And then I mentioned before you need to add a little buffer in order to keep the pH looking good. So you might be wondering, who is going to be doing the 94 degrees, the 60 degrees, and the 72 degrees, and then repeating it, 94, 60, 72, and then repeating it? How would you like it if we needed to repeat this cycling 30 or 40 times? That's kind of a lot of effort and maintaining the temperature in these water baths, but that's the way it was done originally. But we now have a machine or an apparatus or an instrument in the lab called a thermal cycler, and what it does is it is able to change temperatures and cycle between these 3 temperatures. And so what you're going to do is put your little PCR tube inside the thermal cycler, and the thermal cycler is going to automatically jump between very warm, cool, and warm. So hot, cool, and warm, and that'll make up one complete cycle. And so it can actually automatically, once you put this in and hit the start, it will actually do this for as many cycles as you like. And so it's really, really cool, thermocycler. And so we're lucky to have this. Here's a close-up of it. And so in the test tube, over here, like this, it's going to be a little tiny tube, and we're going to be placing that inside there, right there. And so inside this tube, if you recall this, number 1, you would have your template DNA. Number 2, you would have your DNA polymerase, your Taq polymerase. Number 3, you would have a primer. Now, I'm going to say primers because one's in the forward direction and one's in the reverse direction, so you need to have primers. Then you need to have your nucleotides, so those are DNTPs. Those are your A, T, C, and G's. And then you're going to need, if you can guess this, buffer. And then you're going to need a little bit of magnesium chloride. And so you put all of this into the thermal cycler, and the thermal cycler is going to rotate between 95 degrees, 60 degrees, and 72 degrees. And that's going to complete one cycle. Pretty cool. And so basically, DNA, PCR is DNA replication like crazy in a test tube. And I put a picture here of the hot springs of Yellowstone to show you that we got the polymerase from Thermoaquaticus, and it makes millions of copies of the template DNA. It's really cool. And then this is a great schematic, and here's your template DNA, your target area. Now this genomic DNA, if I can do this, this is coming from, in this particular experiment, it's coming from a virus. So that's what we're getting. But we don't want to amplify the whole virus. We want just that little target sequence. And you're like, how are you going to do it? Well, first thing you're going to do is heat it. And so the DNA breaks the hydrogen bonds. Then the little primers, the primers also need to be opened up by the heating. And then they attach right here, both sides. And so this is the cool temperature. And then the warm temperature is DNA polymerase will extend. So these are the three steps in one cycle. Open, attach, elongate, or denaturation, annealing, and extension. So after one cycle, there's 2 DNA. Then after the second cycle, there's 4, and after the third, there's 8. So it's an exponential increase of the target DNA. It's pretty impressive. And so here's a close-up of that. So you're going to need to heat it. This is the one little area, and the heating it again causes it to open. The primers then attach, cool, and then they elongate. And so you get 4, and then you get 8, and it continues on. And I like this slide because in case you're wondering, when the very first cycle, if you're going in one direction, you're going to replicate the whole piece of DNA. You're going to go beyond the template, the target. And over here, you're going to go beyond the target. And over here, you're going to go beyond the target. But eventually, the more cycles you do, you'll just be able to do the target, and then the target, and then the target. And then look at this, after 6 cycles. Can you imagine after 30 cycles how many pieces of DNA that you'd get? And so this is the region that we're interested in amplifying. And so here's my question. So if all of this is happening inside of a test tube, inside the thermocycler, how do you know that this is working? How do you know, like for example, here's the genomic piece of DNA. How do you know that you're getting that one little area for study? Well, I may have mentioned to you before that this one little piece is 500 base pairs. So we should have millions and millions of copies of this 500 base pair. And it's like, well, how would you know that? Well, then you need to pipette that into an agarose gel. Let me just do this. Here's your agarose gel, SkyCam. So if you were to pipette this into the gel like this, put some loading dye in so you can see it. There it is. And then turn the power on like this. And it's like, well, how would you know that it's 500 base pairs long? Well, we have something called a base pair ladder where we could put in lane 1. This is 1,000, 900, 800, 700, 600, 500, 4, 3, 2, 100. If when we load this, 1, 2, 3, 4, 5, if we get lines like this in our gel, this line right here at 500 base pairs will be that. We'll know that this is what's happening. And so, again, this is how you would determine the difference between individuals if you were doing DNA fingerprinting. Of course, you would need to then add a restriction enzyme in order to cut it up so you can see different banding patterns. But it's the first step in amplifying DNA PCR. I hope you're enjoying that. And then finally, let me conclude with this video right here, which I find to be really good. Let me increase it right here. And so it's a great animation. So here's your template DNA. And so what you want to do is notice how the temperature is rising. It's stressing the molecule out. Look out, it's really hot. It's boiling. That's going to cause it to denature. So the DNA then opens up. And you're like, okay, that's good. Then you take the temperature down really cold. And what's going to happen? The primers are then going to anneal to this area. And so they stick there. Now, nothing's going to happen. You need DNA polymerase. And so you take the temperature back up to a warmer temperature. And what that's going to do is allow the DNA polymerase, Taq polymerase, to attach. It's going to grab the DNTPs, which are not shown, and it's going to extend at 72 degrees. And there you have it. That's one cycle. So then you repeat it. It gets hot, and then the molecule opens up, and it gets cold, and then the primers attach. And then it gets warm again, and then the DNA polymerases attach and elongate. And then look at this. After a couple of cycles, you're getting an amplification of just that particular target region, and it continues over and over and over again. And then again, you know that you're successful if you then take your PCR product, which has all of these areas that you're interested in, and then you run a gel, and then you're able to see whether or not you're successful. So I hope you enjoyed this brief discussion on polymerase chain reaction. And if you're following it, that's great. But the truth is, experience is the best teacher. And we're going to actually do this in the lab, and we're going to do it actually a couple of times over the years here. And so I hope you enjoyed it, and thanks for watching.